Okay, in this video we're going to talk about membrane potential. Membrane potential refers to an electrical potential energy that's produced by the separation of oppositely charged particles across the plasma membrane. So kind of to decipher what this is saying is essentially the plasma membrane separates two different fluid environments, the intracellular fluid and the extracellular fluid. What we find is that there are typically more negatively charged particles in the intracellular fluid with respect to the extracellular fluid. And because there's a separation of charges, we call this a voltage, or the membrane potential. Now, cells that have a charge are said to be polarized, so we see that there's a negative side of the plasma membrane and a positive side of the plasma membrane. And because of this difference, there's a polarization there, and a potential difference is referred to voltage. So when you have two different electrical charges that are separated, that is a voltage. So in kind of an odd way, the plasma membrane contains a voltage because it separates oppositely charged particles. Now this voltage only occurs at the membrane surface. It turns out that the fluid farther away from the membranes is relatively neutral, so that membrane voltages within the inside of the cell range from about -50 to about a -100 millivolts. Now the negative sign here just refers to the relative negativity with respect to outside the cell. So we find that the inside of the cell is relatively more negative than the outside of the cell. And this difference in charge, remember, is voltage. And we also refer to this as a membrane potential because there's the potential for charges to move. Remember, oppositely charged particles are attracted to each other. So that's why it's a membrane potential. And that separation of charge means it has a voltage. So what this slide here is showing is basically a pictorial view of this resting membrane potential. Remember, we have the plasma membrane here, and it separates two fluid environments, the extracellular fluid and the intracellular fluid, or cytoplasm here. Now, it turns out that in most cells of your body, at rest, when the cells aren't actively communicating with electrical currents, it turns out that the inside of the cell is relatively more negative with respect to the outside. And this is due to a variety of reasons. For one, there's a lot more protein on the inside of your cells than outside, and typically proteins are anions, so they carry a negative charge. So we find that these negatively charged anions kind of help contribute to the inner negativity of your cells. We also find that because of these potassium leakage channels, they're facilitated diffusion channels, but they allow for positively charged potassium to leave. And what we find here is that because you're removing positively charged ions or cations, once they leave the cell, you're making the inside of the cell relatively more negative by removing positive charges. And once those positive charges end up on the outside of the cell, it makes the outside here, the extracellular environment, relatively more positive as well. One of the reasons why the outside of the cell is also relatively more positive is you have a lot more positively charged sodium out here, and this helps contribute to that positivity. Now it turns out that these charges, you might say that sodium ions can line up here near the plasma membrane, and these opposite charges are sort of magnetically attracted to each other across the plasma membrane, so they line up along the outer and inner leaflets of the phospholipid bilayer. And we refer to this potential difference as a voltage. So if we refer to the inside of the cell having a particular voltage, we find that it ranges from about -50 to about -100 millivolts. In this example, it's just saying that this is about -90 millivolts. And there's different ways to calculate this, but the degree of voltage just tells you about the degree of separation of charge, right? So if you have a very negative voltage inside the cell, it just means that there's a lot more negative charges inside with respect to the outside. Or if it's less negative, like let's say closer to 0 millivolts, we're going to say that there's approximately equal distribution of charges, across the plasma membrane. So there's less potential difference here. But what's fascinating is that we refer to this as not just an electrical gradient, but there's also a chemical gradient. So yes, there are positively charged ions out here that contribute to the positivity in the extracellular fluid, or negatively charged ions that contribute to the negativity in the intracellular fluid. But we also have to take into consideration that there are certain concentrations of ions outside the cell and inside the cell, and this could change how these move. Now, this will become more important when we come back to how neurons function, or brain cells function, and how muscles function later on. But it's important to note that resting membrane potential is essentially the separation of charged ions across the plasma membrane, and we call that potential difference voltage.